Hi, this is Mr. Anderson, and welcome to the Chapter 8a podcast. I'm going to talk about mitosis, and that's how one cell makes an exact copy of a cell. One cell makes an exact copy of a cell. One cell makes an exact copy of a cell. One cell makes an exact copy of a cell. Hi, and welcome to the Chapter 8 podcast. This is on mitosis. Chapter 8 is kind of broken down into two parts. The first half is mitosis, in other words, how cells make exact copies of themselves. And the second one is on meiosis, how they make sex cells or gametes. If we look at this picture right here, we'll look at these under the microscope. But these are onion roots that are in the process of dividing. And so what you can see are the chromosomes. And if you look at the word chromosome, what it really means is colored things. They didn't really know what they are, but we know quite a bit about them right now. And so as I look at this, I can see cells that are in, this would be anaphase right here or metaphase right here. But most of the cells are actually like this and this and this are in a phase called interphase. Now, as we go through this chapter, people have a tendency to get too far ahead and want to get into mitosis and talk specifically about what are the different phases and learn that. And I think you can miss the context. And so first of all, let's start with the idea that not all cells do mitosis. And so this right here is a bacteria. So this is like an Escherichia coli bacteria. Remember, their DNA is a little different than ours. It's just a single loop of DNA, and they've got a cytoplasm or cytosol around that. They don't have a nucleus, and so their DNA is kind of organized in that nucleoid region. It's also a loop, and so their DNA is in a single loop, and ours is in straight linear segments called chromosomes. So it's organized in a little bit different way. So theirs is just a single loop. Now, how do they divide? They don't do this process of mitosis or meiosis. They don't have sex or any of that. We'll get to how they get genetic variability a little bit later. But the way they divide is they simply copy their chromosome. So they'll copy this DNA, and then the cell will start to divide in half, and it eventually will split in half. And we call that binary fission. And so each of those are identical. If there's no mutations, each are identical to that parent. And so a good trick question we like to always ask on the test is, what phase of mitosis is this bacteria in? And the answer is they're not. They just reproduce through binary fission. And so let's get a little bit to the circle of life before we get to the phases again. This is that circle of life. And so I put some clothes on our guys from the text, just because this video might go anywhere. But the way that the life works is that males and females will produce eggs and sperm. Those have half of the genetic information that adult has. And so the adults have 46 chromosomes, so each of these have 23. Those will then fertilize each other to make a zygote, so we return to 46. And so the first podcast on mitosis deals with this, how we go from that first zygote to an adult. And the way we do that is through mitotic division, or the cell cycle. The next podcast will deal with meiosis. How do we go from an adult, which is diploid, towards these gametes? And each of those are a little bit different. As we look at the cell cycle, the book talks about it as it's kind of like the cycle on a washing machine. So you've got your time where it's filling it up, it's adding the suds, it's doing all the dishwasher or the washing machine stuff. And so cells kind of go through a similar cycle. And so they've got three phases of interphase. And so most of the time a cell is alive, it's in this, which is called interphase. And during interphase, a cell will go through G1, where the cell grows. It'll then enter into the S phase, where the cell continues to grow, and then it will copy all of its DNA. And then it goes through the G2 phase, where it continues to grow again. So mitosis is a really small little part of that. And so a better way to think about mitosis is it's simply division of the nucleus. Then we've got cytokinesis. That's where the rest of the cytoplasm divides in half. And so cells can keep going through this whole process, G1, S, G2, and then they'll divide like that. And so that's how that zygote goes from that first cell to all the cells in our body. We'll talk a little bit about this later, but the idea that even though the DNA in each cell is exactly the same, they actually express different genes to make different cells. And so most of the time a cell is actually in interphase, so I want to start with that. What does a cell in interphase look like? Well, its nuclei, so we're looking at its nuclei right here. It'll have a nucleolus, but most of those chromosomes are loose throughout here in a molecule called chromatin, which is essentially DNA wrapped around these histone proteins. And so all of this time a cell is alive, that DNA, remember, is creating RNA, which is making proteins, which are making you the way you are. And so cells aren't going through mitosis all the time. They're actually doing what a cell does. They're making proteins and allowing that cell to operate. Another thing that people usually don't understand about chromosomes is they think they always look like this, like an X, and that's totally not true either. What they look like is that loose chromatin we saw on this last slide. And so the only time a chromosome looks like this is when it's dividing. And so it's gone through the G phase, the S phase, and it's made an exact copy of itself. And so one other term that I really want to explain is homology, or homologous chromosome, excuse me. So you get a chromosome 1 from your mom, and you also get a chromosome 1 from your dad. And so you have a chromosome 1 from your mom, a 1 from your dad, a 2 from your mom, a 2 from your dad. And so that's how we get the 46 chromosomes. And they die exactly the same way. This is a karyotype. You can see that this person actually is a boy because they've got an X chromosome and a Y chromosome. So after S phase, where they've copied the DNA, you have what are called sister chromatids. And so this side is one chromosome. We could think of this as chromosome 1, but it's made an exact copy of itself on the other side. So this is a duplicate of this. So this is exactly like this. Now in the homologous chromosome, this has the same genes in the same location, but they're different copies of those genes. So now we finally got to the meat of this. So how does it actually do this? And so after those chromosomes show up, so they're now in prophase, this is my way to remember that, interphase, prophase, metaphase, anaphase, telophase. During prophase, the chromosomes actually will coalesce, and so we can see them. Then our book calls it prometaphase, but in prophase, then the nuclear membrane will start to break apart. These centrosomes will go to either side, and those are made up of microtubules. You'll be able to see the chromosomes during prometaphase. The nucleus, or the nuclear envelope, kind of dissolves. They'll then line up or meet in the middle during metaphase, so they'll line up right across the middle. Their centromeres will be attached to this spindle, and they'll line up in this perfect straight line across the middle of the cell. And then finally in anaphase, they'll be pulled apart to either side. So you can see the chromosomes being tugged either in this direction or this direction. And then finally we have telophase or telophase, and in that a new nuclear membrane will form around each of these. Now you have a nucleus, but we're not done yet because we have to divvy up the rest of the cell. And so we then go through a process called cytokinesis. And what happens in that is that the rest of the cytoplasm will split in half. And the reason that's important is that there's a lot of stuff in here that we need. So mitochondria, for example, get passed from generation to generation and from cell cycle to cell cycle through this cytokinesis. Cytokinesis is a little different in animals and plants. In animals, since they don't have a cell wall, a cleavage furrow will form right down the middle. It's two proteins that do that, actin and myosin, and then it'll just pinch it shut, kind of like a drawstring, and you have two cells. In a plant cell, you'll form what's called a cell plate in the middle, and then we can see it right here. A cell plate will form as we tow little bits of the cell wall using vesicles to the middle, and then eventually you have a new cell wall form in the middle. And so this would be cytokinesis in a plant. Now cells will continue to grow as long as they have nutrients and a place to expand into. And so if you put cells in culture, so these are cells just growing in, think of it like a petri dish, they'll continue to grow until they cover the whole bottom of that. And that's because they get signals from around them, and those are called growth factors, and those growth factors say, on the protein surface, say, hey, it's time, you can now go from the G1 phase to the S phase and go through mitosis. But until you get those growth factor signals, they don't continue to grow. And so cells of, for example, your brain are stalled in what's called the G0 phase. They're never going to divide. And so the number of neurons you have at the beginning is going to be equal to the number of neurons you have at the end. And so sometimes cells lose the ability to control that copying, and so they keep copying over and over and over, and eventually those cells become cancerous cells. And that's bad because they can cause other cells in your body to become cancerous as well. So a little bit in review or overview, the process of mitosis, that's what we're dealing with here on the left side, is taking a cell, in this case it's one that's 2n equals 4, and making two exact copies of that. And in the next part of the chapter we'll talk about how you can take this cell and make stuff that's way different. So I hope that's helpful.